I'd like to show you a comparison of mitosis and meiosis. Mitosis, the purpose is to create more of the same type of cell. So in your body, the purpose would be to create more liver cells or more stomach cells. Meiosis is to create the sex cells or the gametes. Mitosis, because we want just more of the same, we're going to start with a cell called a parent cell, and we're going to end with daughter cells that are identical to that parent cell and to each other, of course. In meiosis, the purpose is to cut the number of chromosomes in half. That way, that sex cell can join with a sex cell of the opposite gender to create a whole, or the very first cell of the offspring, called the zygote. So what I'm going to do is I'm going to play mitosis, then I'll play meiosis, then I'll go back and I'll kind of step through each carefully, and then I'll go through it a third time and I'll add more pieces of the puzzle. Here we go. I'm just going to let this play out. Mitosis. And meiosis. The most obvious difference, of course, is that we started with one cell on each side, and we ended with 2 cells over here, and we ended with 4 cells over here. What you also should notice is that I count 2 chromosomes in each of these daughter cells over here on the mitosis side, and I count only 1 chromosome over here on the meiosis side. Finally, you probably noticed that on this side, we started with a parent cell that had a red chromosome and a blue chromosome, and we ended with daughter cells that have each a red chromosome and a blue chromosome. Over here, we get kind of mixed up chromosomes. Recombinant chromosomes, we tend to call those. So, let's start back over and see how all of that occurs. I'll start on the mitosis side. Notice again, we have a single parent cell, 2 chromosomes. Over here, same type of parent. First thing we're going to do, we're going to start in what's called G1, a cell that is never going to divide, or hasn't received a signal yet to divide, will stay right here. Just stay right here using its DNA to do what it needs to do. Move it to that same phase over here. Notice no difference. Once a cell receives a signal to divide, it goes through what's called the S phase of interphase. So G1, S, and the next phase, G2, are all part of what's called interphase. The cell isn't dividing yet, but in the S phase, it doubles its DNA, it replicates its DNA so that it can split. Let's move into G2. In G2, what happens is the cell is just checking to make sure it can go through cell division. All right, notice no differences. So let's move into the first phase of mitosis. There is one thing that is not being shown, well, a few things that are being simplified. First of all, in interphase, G1, S, and G2, you wouldn't actually see the chromosomes yet because they're all unwound, so that the cell can use them and can make a copy of them. It's easier to make a copy when they're decondensed. These are just really compact spools of DNA on both sides here. So the other thing is that DNA would be contained inside of a nucleus in both cases. But they're not showing that just for ease of animation. So we move into what's called prophase. That's where the nucleus, the nuclear envelope, would be broken down on both sides. Now, I'm going to stop it right there. So far, it doesn't look too different. on the left, we're in prophase. On the right, we're in what's called prophase 1. We had to go through 2 cell divisions in order to get to those 4 cells. 1 division led to 2 cells. 2 divisions leads to 4 cells. So in the first prophase, that's where many of the differences between mitosis and meiosis occur. So in the first prophase of meiosis, what actually happens is that pair of chromosomes is going to find each other. They're going to come very close together. They're going to do what's called synapse, which really just means to connect. And then they're actually going to swap pieces of DNA. That's called crossing over. That's how we wound up with those chromosomes that were kind of a mix and match of red and blue in the end. That's prophase 1 over here. We also will form that spindle. Then that spindle helps to separate the chromosomes from each other in both prophase. In both mitosis and meiosis. So we'll continue to make that over here. We move into what's called metaphase. The spindle is actually connected to the chromosomes on both sides. The animation just isn't showing that. Notice how they're lined up. In metaphase of mitosis, each individual chromosome, each individual replicated chromosome is lined up by itself. Over here, the chromosomes, the replicated chromosomes have found each other and they are paired up together. So then when we get to anaphase, what's going to happen is the replicated chromosomes are going to get pulled apart. This is considered to be a chromosome with 2 chromatids. Now what happens is we have unreplicated chromosomes, or chromosomes with 1 chromatid each. This is still a chromosome, and this is still a chromosome. Over here, what we're going to do is we're actually going to pull that pair of chromosomes apart. Right, we're going to pull the chromosome pair apart all the way to the end. So we look pretty different now. Over here, we've got 1, 2 chromosomes on this side of the cell, 1, 2 chromosomes on this side of the cell. Over here, we've got 1 chromosome on this side of the cell and 1 chromosome on this side of the cell. This is a chromosome, but it's replicated, made of 2 chromatids. This is a chromosome right here, unreplicated, made of 1 chromatid. It's important that over here in mitosis, you don't try to put those 2 chromosomes together. They will not come together. All right, so then I can just play this out. That's cytokinesis. We split this cell. This cell is identical to this cell. They each have 2 chromosomes, 1 red one, 1 blue one. And they are each identical to the original cell. If you remember, that original cell had 1 red one, 1 blue one. Let's keep going over here. We'll split the cell. Instead of having 2 chromosomes per cell on this, like we do on this side, we have just 1 chromosome per cell. Again, it's okay that it's made of 2 chromatids. That's 1 chromosome. This is 1, 2 chromosomes. All right, let's keep going. Hopefully you recognize kind of what happens here. In prophase, we start making that spindle. The second prophase, we call it prophase 2. We're going to line the chromosomes up in the middle. Of course, since there's only 1 chromosome here, it doesn't look a whole lot different, but they're lined up individually along the middle, just like they were over here in mitosis. We just line up each individual chromosome. It's just that we have half as many. And then we rip each one apart. Again, now this is called a chromosome. It's just an unreplicated chromosome, or a chromosome with just 1 chromatid, made of just 1 chromatid. And this is another chromosome. I know that for a lot of people that math just doesn't seem right. I split something in half and now I have 2. No, I should have 2 halves. No. This, oops, I guess it just decided to kind of go to the end there. There we go. We wind up having now 1 chromosome per cell. But they are all different from each other. So the last thing I would like to do is talk a little bit about a few more terms. We've got over here on both sides, we've got a diploid cell. I see at the end of diploid cell over here. Diploid means we have homologous pairs, or every chromosome has a pair, a matching chromosome. They have the same genes in the same order. They might not have the same versions of those genes. So over here, let's just say we find the gene for earlobes. Well, gene for earlobes right here. But maybe on this one says attached earlobes, and this one says free earlobes. Maybe down here we have the gene for earwax, and over here we have wet earwax, and over here we have dry earwax. And they don't have to be different, they can be the same, but they have the same genes in the same order. That cell is diploid, and this cell is diploid. Now, if I step through quickly, just going to get all the way to G2. Now I'm going to line them up in metaphase, go through prophase all the way to metaphase. And let's go ahead and just pull them apart. So on this side, we rip each chromosome apart into unreplicated chromosomes. And over here, we're going to pull the pairing apart. Let's split the cell. Notice over here, I still have a pair. I still have a red one and a blue one. That's still a diploid cell. Over here now, though, this chromosome's pair is over in this cell. We have separated the pairs. So this is called a haploid cell. I only see one chromosome from the pair. Your cells start with 46 chromosomes rather than just 2 like we did here. But there are 23 pairs of chromosomes. So this cell would still have 46 chromosomes if it were your cell. This cell would have 23 at this point. When we split each of these chromosomes apart, we rip those chromatids, separate those chromatids from each other. Remember, that doesn't change the fact that we still have it doesn't change the number of chromosomes. So this is a chromosome. We rip it in half, and now there's 2 chromosomes. So each of these daughter cells that we have now has 1 chromosome. The parent cell started with 2 chromosomes, a blue one and a red one. Now I've got 1 chromosome. And it's kind of a mix of both.